student at NC State in the plant biology department, and we're really excited to share our research with you. So I first would like to introduce you to my interns. This is Connor, Corey, Sydney, and Delaney, and they have been instrumental this summer in helping me do my research. And so I'm really excited to be here with you to share what they've been doing and some of my overall research goals. So to start out, a little bit of background information. We're focusing our research in oat. Oat is a very complex crop. It's got a complex genome, which makes it very challenging to study. It's a hexaploid, so it has 3 genomes, an A, a C, and a D genome. And there are a lot of translocations and structural differences between these genomes, which make it very challenging. But in spite of these challenges, there are a lot of traits that we're interested in that oat has. And so, like you've heard, the goal of P2EP is to find out what plants are making, how they're making it, and how that benefits human health. So the couple of compounds that we're interested in is beta-glucan, 1314 beta-D-glucan, and that has been shown to reduce cholesterol. Another one that we're interested in is avenanthromide, which has antioxidant capacity, which we know is also contributing to human health. And so these are the traits we're interested in. But before we can really dive into those traits, we first had to develop some genomic tools. And so this is the overall arching purpose of our research, is these 3 phases. The first one is to develop genomic tools that will allow us to do association studies and associate genes to traits. And so the next step is to identify these quantitative trait loci, or regions of the genome that contribute to production of these compounds that we're interested in. And the last step would be to identify any candidate genes and then to validate those candidate genes using transformation methods. And so we'll move on to that first step, developing tools. This was a very big project. Because oat is so complex and it has a complicated genome, developing genomic tools is a big challenge. And so we set out to develop a consensus map that would allow us to do association studies. And we developed this consensus map using 12 different populations. And we used the interns a lot to do this. We mapped these 12 populations individually. And then once we had those individual population maps, we then merged those together. And so we had to identify the different homologs, the different homologous linkage groups from each of the 12 populations and merge those together into a single population, which is a huge challenge with such a complicated genome. But we're happy to say that that was published and we came out with a really good result. Here's a kind of a picture of our map. And it was a very dense map with a lot of markers, which was instrumental in us being able to go on to the next step of making associations from genes to our traits. So before we could do the GWAS, we first had to get some study populations and to look at those populations and identify certain aspects of those to make sure that these populations were good populations to use to identify these traits. And General Mills was kind enough to give us some of that data. They provided us with 3 diversity panels: a spring panel, a winter panel, and a world diversity panel. And each of these panels were grown in 2 or 3 locations. And so we had a lot of data to work with, which was great for us. Once we had those panels in place, we first wanted to look at the population structure. We wanted to make sure we were controlling for population structure to eliminate as much of the false positives that we could. We didn't want to have false positives due to different allele frequencies from different subpopulations. And so we used 2 methods, principal component analysis and a relationship analysis, to look at population structure. And so once we had that all figured out, we were ready to move on to our analysis of our traits to identify associations for our traits. And Connor, he actually has been an intern in the program for 4 years, and this is his 2nd year in my group. And last year we worked a lot on identifying QTLs for beta-glucan. And so he's going to share a little bit of that work, and I'll pass it on to him.
Thank you, Scott. And so, as Scott mentioned, my name is Connor Reed. I've been an intern here for 4 years, and I've been working with Scott for 2. And so I've had the unique opportunity to be able to help him with his research for 2 years. And so what I'm going to do is I'm going to answer the first question first of Dr. Lila's 3 questions that we set out to research here on the campus. And that is, what are these plants producing? And so we were looking at mixed linkage beta-glucan. And so mixed linkage beta-glucan is found in the cell wall of oat. And what makes it unique from other cell wall components is that, like cellulose, for example, will have these 1,4 linkages between its glucose subunits. And so these 1,4 linkages come together to make a relatively straight structure. But beta-glucan has these 1,3 linkages, which creates kinks or bends in its structure. And you can see that on the diagram behind me. These kinks are what give beta-glucan its unique properties in the human body. Specifically, beta-glucan is able to interact with LDL, or your bad cholesterol, and has been shown to be associated with a decrease in that bad cholesterol. You guys might have actually seen there's a heart on the Cheerios box that says Cheerios can help to reduce cholesterol, and that's because of the beta-glucan in the oats used to create those Cheerios. Also, that heart goes on to say Cheerios helps reduce cholesterol as well as decrease the risk of heart disease. And that's very important because 1 in 5 people in the United States actually suffer from coronary heart disease. So that's something that we're hoping to impact through our research. So last summer and this summer as well, we ran a genome-wide association study focusing on beta-glucan. And what that means is we were looking to determine the genetic pathway that leads to the synthesis of beta-glucan in oat. And so you can see behind me an example of the output of that genome-wide association study. And so there are a bunch of circles on that diagram, and each circle represents a marker in our data. And these markers are spread out on the x-axis by linkage group, and then within each linkage group, they're subdivided into the location where that oat crop was grown, so that's either Aberdeen, Fargo, or Lacombe. And then on our y-axis, you have the position on the linkage group where that marker is located. And you'll notice that some of the markers are colored. This was the output of our genome-wide association study. The lighter color, the gray-colored markers were not shown to be significantly associated with our trait, which was beta-glucan, while the colored markers were shown to be significantly associated with beta-glucan. And so with the genome-wide association study, there is the risk of finding false positives, and so we wanted to make sure that we didn't identify or define any QTLs, those quantitative trait loci that Scott was talking about, or regions of the oat genome that we're interested in, unless we found significant markers, so those are those colored markers, replicated across locations, so ideally replicated across Aberdeen, Fargo, and Lacombe. And so you can see there are some boxes on this graph. And that's where we found those replicated markers. And there are 2 different colored boxes up there. There are orange boxes, which were just replicated. These markers were just replicated across a single panel. So we had our spring, our world, and our winter panels. But there are green boxes as well. And that's where not only do we find replicated markers across the different locations where the oat was grown, we also found replicated markers in the same position across our different panels, our spring, winter, and world panels. So these were the areas, these green boxes were the areas of the oat genome we were most interested in because we found such a significant association with beta-glucan. So once we'd done that, we now wanted to know, well, what gene is located in these QTLs? And so we know there's a gene called CSLF6, which is known to produce beta-glucan in other similar crops. And so one of Scott's colleagues was able to map CSLF6 to a marker, and we looked at our QTLs that we identified on that last slide, and we found that one of the genome's copies of CSLF6 actually falls on our map. So this made us pretty confident that CSLF6 was the gene that we're looking for. We went on to verify this by looking at 2 of our other QTLs, 2 of those other green, very highly significant QTLs, and we found that they were fairly similar to a region of the barley genome that contained barley's copy of CSLF6. So just providing even more confidence that CSLF6 was the gene that we're interested in. But as Scott mentioned, oat is hexaploid, which means that it's not just going to have 1 copy of that CSLF6, it's going to have 3 copies of CSLF6 for each of its 3 genomes, the A, the C, and the D genome. So now we were interested in figuring out, well, how do these different genome copies of this gene, the CSLF6, impact beta-glucan production? So I'm now going to hand this off to Corey, one of the other interns this summer, and he's going to talk to you about the C-genome copy of CSLF6.
Thank you, Connor. So above are graphs that depict past research done on the C-genome copy of the CSLF6 gene. The bottom 3 graphs show a negative correlation between the expression of the CSLF6C genome copy of the gene and beta-glucan. This means that with an increase in the expression of the CSLF6 gene and the C genome, there's actually less beta-glucan expressed. In order to better understand the genetic pathway that's involved that causes this, we are going to transform Arabidopsis, a plant that naturally contains no beta-glucan, with both the A and C genome versions of CSLF6. And we will, hopefully from the data, we will glean information that will tell us more about why the C and A and D genomes act the way they do on beta-glucan production. Now I will hand it off to Sydney, who will tell you about avenanthramides.
So this summer, we started doing a genome-wide association study on avenanthromide. And the reason that we were interested in this is because it is exclusive to oats and is not found in any other of the different crops that we've been looking at. And the reason that it's so interesting is because it is known for antioxidant capacity, and it is also associated with the reduction in the chance of atherosclerosis. And if we can help with lowering that, then it actually will lead to an overall healthier heart. This figure here is a genome-wide association study. Is a genome-wide association study that we conducted this summer. Along the x-axis on the bottom, you have the location that the crop was grown, and then you also have the linkage groups. And along the y-axis, you have the position within the linkage group. And I'd like to draw your attention to that circle on the 2 markers on the top there. And those 2 markers we are very interested in because they show a high level of correlation with the levels of avenanthromide content in oats. And now that we have these markers, we can move forward with our future plans, and I will pass it along to Delaney, who will tell you a little bit more about that.
So as Sydney said, we identified 2 locations on the oat genome that are associated with avenanthromide production. However, because the oat genome is not yet sequenced and no other crop produces avenanthramides, we're not yet able to identify the exact genes at this location. As far as beta-glucan goes, we also found a location for the oat genome that's highly associated with beta production, and we do know the gene CSL6 at this location. So with these 2 findings, we are able to give it to oat breeders, who will then be able to breed for oats that have higher beta-glucan and avenanthromide production. This will build stronger oats and will make our cereal